at my research. Today I'm going to tell you about a pooling-based approach to mapping genetic variants associated with DNA methylation. I'll begin by telling you a little bit about why I'm interested in DNA methylation. I will then describe the previous method for identifying variants associated with methylation. After that, I will describe our novel method for identifying such variants, and I will then tell you about some of our results. So what is DNA methylation? One of the bases of DNA that we have is a cytosine, and cytosines can be either methylated or unmethylated. And by methylated, I just mean that they have a methyl group attached to them. So unmethylated cytosines can occur before any base of DNA, but methylated cytosines almost always occur before guanines. We will abbreviate a cytosine followed by a guanine as a CpG. CpGs often do not occur in isolation, but instead occur in regions with many CpGs, which are called CpG islands. Because of this, many methylation studies have focused on methylation patterns that occur only in CpG islands. As I will show you later in our talk, it may be worthwhile to study methylation patterns outside of CpG islands, which is something that our novel method enables us to do. The reason I'm interested in DNA methylation is that DNA methylation can play an important role in transcription. If a promoter of a gene is highly methylated, that gene tends to have low transcription. If an intragenic region of a gene is highly methylated, that gene tends to have high transcription. If a distal regulatory element that is involved in activating a gene, such as an enhancer, is highly methylated, the gene tends to have low expression. However, there are only a few examples of methylation patterns and enhancers affecting gene expression in the literature, and this is because many studies of DNA methylation have focused almost exclusively on methylation in promoters and inside of genes. As I will show you, our method enables us to also study methylation patterns in more distal regulatory elements. So methylation has been shown to be a heritable trait. There are a number of studies that have demonstrated that it can be passed on from generation to generation. Because of this, it is interesting to study genetic variants that are associated with DNA methylation. So if we have an allele of a SNP that is highly correlated with the methylation status of a nearby CpG, which by methylation status I just mean whether or not that CpG is methylated, we call that SNP a methylation quantitative trait locus, which we abbreviate as MQTL. Because methylation can play an important role in transcription, MQTLs can also have dramatic effects on transcription. All previous MQTL mapping methods that I know of to date have used the technology for assaying methylation called methylation microarrays. In a methylation microarray, we select a subset of CpGs of interest, and we probe the methylation status of these CpGs. We do this separately for every individual in our study, so what we obtain is an average methylation status for that individual. We also then do a separate genotyping experiment for each individual, where we select a subset of SNPs of interest and identify the genotypes for each individual of those SNPs. While methylation microarrays have enabled us to identify numerous MQTLs, they have a number of drawbacks. First of all, the largest methylation microarrays that have been used study the methylation of less than 2% of CpGs in our genome. Secondly, we have to do a separate experiment for every individual, which can lead to sample variability. In addition, we have to do a separate genotyping experiment, which means we have even more work. And finally, we get an average methylation status for each individual, so we cannot identify allele-specific methylation. So the goal of our study was to answer the following question: which SNPs are MQTLs? We wanted to do this in a novel and complementary way to other studies, because we wanted to be able to assay this genome-wide in a way that we can detect allele-specific methylation in a single experiment. Once we have identified novel MQTLs, we can then investigate the following follow-up question: do these associations provide mechanisms that explain how SNPs may affect transcription or disease? So as Nancy mentioned earlier, many MQTLs from the array studies have been shown to also be EQTLs, meaning SNPs that are associated with transcription. And for these EQTL and other quantitative trait locus studies, we often don't know the mechanism through which a SNP that's associated with the trait may influence that trait. Now, if that SNP we show is also strongly associated with methylation of a nearby CPG, that would suggest that the SNP may influence transcription of the other trait with which it's been associated by affecting the methylation status of the nearby CPG. So, in order to approach this problem of finding MQTLs in a new way that overcomes the limitation of the arrays, we use a technology called whole-genome bisulfite sequencing. Bisulfite sequencing is a sequencing method that, in addition to sequencing each base of DNA, also enables us to identify whether or not cytosines are methylated. Because this is a whole genome method, we can probe the methylation status of every cytosine in the genome. In addition, because this is a sequencing method, we can often separately investigate the methylation status of the DNA from an individual's mother and from the individual's father, enabling us to identify allele-specific methylation. So why is identifying allele-specific methylation so useful for MQTL mapping? In order to understand this, let us consider a heterozygous individual. And this is going to be a similar explanation to one that Roger just showed for ASC and EQTL mapping. So in this individual, if we're using the microarrays, what we would find is we would find a genotype of heterozygous, we'll call a genotype of 0.5, and then we'll also get an average methylation status of 0.5 because the individual is methylated on half of its DNA. So the microarray is doing averaging. In whole genome bisulfite sequencing, what we will see is that we'll have a genotype of T allele, which we'll call allele 1, being associated with a methylation status of being methylated, which we'll call methylation status 1. For the A allele, which we'll call allele 0, we'll see a methylation status of 0 because our cytosine is not methylated. Now, if we imagine an MQTL with this pattern for which all of our individuals are heterozygotes, for our methylation microarrays, we'll see many 0.5s associated with many 0.5s, so we will not be able to detect the association. However, for bisulfite sequencing, we will consistently see 1s associated with 1s and 0s associated with 0s, thereby enabling us to detect the MQTL. Thus, using whole genome bisulfite sequencing, we may be able to detect MQTLs for CPGs that are in the arrays that the array methods were unable to detect because we can detect allele-specific methylation. Now, you may be wondering why previous MQTL studies have used methylation microarrays given all of the advantages I just described of whole genome bisulfite sequencing. The reason for this is that whole genome bisulfite sequencing is extremely expensive. As I will show you, our method is able to cut the cost of whole genome bisulfite sequencing. The way we cut the cost is by using a pooling strategy. Before going into this strategy, I should mention that we applied this strategy to a set of 60 Urubin lymphoblastoid cell lines, which we abbreviate as LCLs. Our reason for choosing these LCLs is that many other studies have been done on the same cell lines, so we can later integrate the data from these other studies with the data that we generate. So the way we do pooling is we take a small amount of cells from each of 60 individuals, and we combine them into a single pool and do bisulfite sequencing on the pool. Now, you may be wondering how we can possibly find MQTLs given that our data is coming from a pool of cells from many individuals. Here's the trick. If a SNP is sufficiently close to a cytosine, there will be many reads overlapping both the SNP and the cytosine. So from these reads, we can directly identify the MQTL. In this toy example, what we see is that our T allele is always associated with a lack of methylation, and our G allele is usually associated with methylation. So in this toy example, we can see from these reads that there is an association between the allele of the SNP and the methylation status of the cytosine, even though we might not know which particular individual each read comes from. The way we quantify the significance of the association is that we count up the number of combinations of each allele methylation status pair, and then we use Fisher's exact test to quantify the significance of the association. So one drawback of this method is that we require that both the SNP and the CPG of interest be on the same read, which requires them to be within a few hundred bases of each other. A recent study using a large methylation microarray showed that the majority of the SNPs that are most likely to be causal are actually within a few hundred bases of the associated CPG. So now that I have shown you our pooling method, I'm going to show you some simulations that we did that demonstrate the benefits of being able to detect allele-specific methylation. For these simulations, we imagined that we had a population of individuals in Hardy-Weinberg from which we can sample many reads. So from this population, we sampled a particular number of reads of interest, and imagined these reads were randomly distributed across our individuals. We then averaged both our SNP, getting our genotype, and our methylation status, and we computed the association for the MQTL from these averages. We called this method the traditional method because it, like the microarrays, is doing averaging, and microarrays have been traditionally used for MQTL studies. We also simulated our pooling method by, instead of scattering the reads across individuals, imagining that all of our reads were in a single pool, and then computing the association for the MQTL directly from the reads. We repeated this process 10,000 times, and we defined power to be the fraction of simulations in which we were able to detect the MQTL. Our simulation results show that the pooling method consistently performs at least as well and often much better than the traditional method. In these plots, our x-axis is the effect size of our MQTL. Our y-axis is power as defined on the previous slide. Each of these plots is for a different number of reads because we tried imagining having different numbers of reads. And our red line is for the pooling method, and the blue line is for the traditional method. What we see is that the pooling method does especially well in comparison to the traditional method for MQTLs with low effect sizes. For example, for 160 reads, the pooling method identifies the MQTL almost 4 times as often as the traditional method. This demonstrates the allele-specific being able to identify allele-specific methylation can help us identify MQTLs that we cannot find by using the microarrays, and that this is especially true for MQTLs where the SNP allele is not perfectly correlated with the cytosine's methylation status. Now that I've shown you the results from our simulations demonstrating some of the benefits of our method, I will show you the results from our empirical data. In our study, we evaluated over 746,000 CPGs, representing the largest number of CPGs evaluated in any MQTL study to date. You may be wondering why we did not evaluate every CPG in the genome, and the reason for this is that we had, on average, 40x coverage for base with a fair amount of variance. Because of this, unfortunately, for many CPGs, there were so few reads covering them that even in MQTL, where the SNP's allele was perfectly correlated with the cytosine's methylation status, would not be able to be identified with statistical significance. So we eliminated those CPGs from our study. From these CPGs, we were able to identify over 2,000 MQTLs with an FDR of approximately 0.06. To show you an example of what an MQTL looks like in our data, we have 2 bar graphs, one for each allele, and on our y-axis we have the number of reads. So what we see here for this MQTL is that our A allele is associated with lack of methylation of the nearby CPG, while our C allele is associated with methylation of the nearby CPG. Now, some of our MQTLs have actually previously been associated with other traits. For example, we found 29 SNPs that have previously been associated with exon-level transcription in Yoruban LCLs, and this is more overlap than we would expect by chance. In addition, we found 34 SNPs that have previously been associated with open chromatin in Yoruban LCLs, also more than we expect by chance. For those of you who don't know, open chromatin is a measure of how accessible DNA is to regulatory mechanisms that might bind to the DNA. Now, interestingly, we found that the allele that was associated with methylation was almost always associated with a lack of open chromatin. This is not surprising because DNA that is methylated is generally less accessible. In addition, we found MQTLs that have previously been associated with diseases in genome-wide association studies. We had 5 exact overlaps with GWAS hits, including 2 that we thought were particularly interesting. One is a SNP that has been associated with age-related macular degeneration in multiple studies, and the other is a SNP that has been associated with the ratio of visceral adipose tissue to subcutaneous adipose tissue, which is a measure of obesity. I should mention that neither of these MQTLs were found in previous studies, and the reason for that is that the CPGs with which they are associated are not included in any of the arrays. I think that the arrays neglected to include these CPGs because these CPGs are outside of CPG islands. This demonstrates that it may be beneficial to study methylation of CPGs outside of CPG islands. To show you what these MQTLs look like in our data, we have a plot for macular degeneration, which is our first MQTL, and the MQTL associated with obesity, which was the second one. The allele boxed in red is the risk allele for the disease. What we see here is that for the MQTL associated with macular degeneration, the risk allele is associated with lack of methylation of the nearby CPG, or for the MQTL associated with obesity, the risk allele is associated with methylation of the nearby CPG. We decided to validate these MQTLs using pyrosequencing. Pyrosequencing is another assay for measuring DNA methylation that, like the arrays, computes an average methylation status for each individual, but is thought to be much higher quality than the arrays. What we found is that the pyrosequencing results confirmed what we had found using our whole genome bisulfite sequencing. This suggests that these MQTLs are real MQTLs and not just some artifact of our whole genome bisulfite sequencing experiment. In conclusion, we provided novel pooling and association methods for MQTL mapping with increased power for proximal MQTLs. We used these methods to identify over 2,000 MQTLs, over 99% of which have not been identified using the arrays. Finally, we found MQTLs that are also associated with transcription, open chromatin, and disease, which suggests a mechanism through which these SNPs might affect these other traits. I would like to thank my advisor, Hunter Fraser, our experimental collaborators in Michael Kober's lab at UBC, Sarah Ma, who did the bisulfite sequencing, and Julie McIsaac for the pyrosequencing, the Fraser, Kohler, Kober, and Kundaje labs for useful discussions, and the DOE, NSF, and Stanford for funding. If you're interested in learning more about this project, feel free to come by my poster, which is poster number 42, and I'd be happy to take any questions.
Thanks, Irene. Very interesting talk. This might be a naive question, but I was wondering if there is any value in being able to identify your EQTLs on an individual by individual basis, and that I presume could be done by probably multiplexing your whole library at the cost of losing some depth. So,
right. So yes, we could do very low coverage bisulfite sequencing where we barcode each individual. However, there are a number of drawbacks to that. The main one being this lead can lead to sample variability. So if we can't do all of the individuals in the same day, we might then have batch effects. And it's also a lot of additional work. So this is a much easier method.
Gotcha.
So yeah, the benefit to that would be that we could identify more distal MQTLs because we would actually know which individual each read comes from.
Right, and a quick question. What's the percentage of your EQTLs that you found, MQTLs that you found that are outside CPG islands?
That's a good question. I don't remember that, but good question.
So I was delighted that you're doing this now because, as you know, I'm sure many of the methylation changes that occur when you turn a gene on are actually in the shores and shelves,
yes,
not in the islands.
Yes.
So that's really good. I just wanted to bring up the fact that because methylation changes are actually induced by transcription factor binding,
yes,
that means that your lymphoblastoid cell lines are really only telling you about genes that are differentially regulated in lymphoblastoid cells.
Yes.
And I think going back to where Nancy started this morning, you obviously will want to extend this to a much broader range of tissue-specific samples. So is that anywhere near feasibility?
I would love to see this done in primary tissues. We currently don't have access to the kind of enough cells that we would want in order to do this because we'd like to do this very deep in primary tissues and be able to identify even more. But I would love to see that happen.
Yeah. But they need a lot of sample. They need a lot.
We need to sequence extremely deep, so we need very large.
So a couple of beautiful work. A couple of questions, technical questions on the read length. So I mean, I presume that your reads are fairly short.
So our reads are 101 base pair paired end.
Okay. So does the pairing allow you to map them?
Yes.
Okay.
So we were able to identify a number of MQTLs that were a few hundred bases apart.
Very cool.
Because of the paired end.
Now, what fraction are in the pairs rather than on the same read?
The majority are in the same read.
Very interesting.
But
so, given the clustering that you pointed out, I'm just wondering if any of your SNPs are perturbing not the methylation mark that you care about, but other methylation marks at that exact location.
So that is a possibility. We actually excluded CPGs with SNPs from our study and SNPs that are in CPGs because the way bisulfite sequencing works is that it converts these that aren't methylated to thymine. So if there's a CT SNP, then we had to exclude that. So it is possible that that is happening, but that's something that is hard to find, that we can't really find using any bisulfite sequencing method just because of the way it works.
And then the last question is, given the large number of methylation of CPGs that you're interrogating, I was surprised that you only found so few
So a lot of
methylQTLs.
this, I think, was because of our lack of sequencing depth.
Okay.
So even though, we said, okay, the criteria for a CPG that we included was having enough reads that in MQTL, where the SNP's allele was perfectly correlated with the CPG's methylation status, we'd be able to find with statistical significance. Which means that if we didn't, for our CPGs with lower coverage, if the correlation wasn't quite perfect, we wouldn't be able to find it. So I think with more sequencing depth, we'd be able to find a lot more.
I presume that it's just a question of money. It's not a question of sample size, right? Your libraries have much more complexity than what you're sequenced at right now, right?
So I think we ended up using up most of our libraries
I
for
see.
this.
Okay, so you would need more sample.
Yeah.
Okay, great. Thank you very much.
Thanks.
Okay, let's thank Irene again.